Hello, my name is Butler, and I am a junior at Untitled High School. And my project name is Curing Cystic Fibrosis with CRISPR-Cas9. Now, what is cystic fibrosis? Cystic fibrosis is a single-point mutation, meaning that only one letter of the DNA ladder changed. This causes defective protein to be created and not control water movement in the airways of the lungs. This causes a buildup of mucus that provides a breeding ground for bacteria. In the next couple of slides, I'm going to speak on 2 previous attempts on curing cystic fibrosis. Here, the common cold virus was employed to send the correct form of DNA into the patient, but this caused inflammation in the lungs because the virus was too strong. Also, a flaw in this design was that the original DNA of the CF, cystic fibrosis, was still there and still had some effects. Here's an attempt at using liposomes. This took advantage of the nature of cell membranes. The cell membrane brought the liposome into the cell and then extracted the DNA from that liposome and then sent the DNA into the nucleus, hoping that that DNA would be brought into the double helix. But the original gene was still there for cystic fibrosis, so it really only hid the side effects for a couple of days. In the next 4 slides, I will explain what CRISPR-Cas9 is, where it came from, and how it works. As I said before, CRISPR-Cas9 is an enzyme RNA complex. The RNA guides it to the incorrect DNA so that the enzyme can edit it and change the one incorrect nucleotide that caused cystic fibrosis. Here's the team that discovered CRISPR-Cas9. It was found in a species of bacteria in archaea, and CRISPR-Cas9 was used for the bacteria in archaea to resist viruses and plasmids that could potentially be deadly to the bacteria in archaea. Now, how would this be delivered to the nucleus of the diseased cells? This would be through the employment of the adenovirus. Here is a figure of the adenoviral life cycle. The adenovirus will attach itself to the cell, then injecting the hexon of CRISPR-Cas9 into the cell. This will be sent to the nucleus where CRISPR-Cas9 will then use its DNA editing properties to change the DNA of the patient. When the RNA guides CRISPR-Cas9 to the incorrect DNA, Cas9 will delete the incorrect section of DNA and transcribe the correct form of the DNA from the RNA sequence, then bonding it to the DNA, resulting in a correction of the DNA, therefore getting rid of the disease. Here are a couple of animal models. This CRISPR-Cas9 design was used to cure lab rats of liver disease. It was also used to go through targeted mutagenesis in nematode worms. And it also was used to permanently turn off gene expression in cultured human cells. Here is a proposed clinical trial. A group of CF, cystic fibrosis patients, consisting of children, teenagers, and adults, will be treated with CRISPR-Cas9 through a nasal spray. This guarantees a smooth transfer to the lungs. The CRISPR-Cas9 virus will attach itself to the infected cells, and then the CRISPR-Cas9 will then be delivered to the nucleus of the cells. The CRISPR-Cas9, after arriving in the nucleus, will then edit the incorrect DNA, and as a result of the treatment, 1/3 of the cells will be corrected, resulting in a permanent cure of cystic fibrosis over time. I would like to give special thanks to Ms. Sherry Carlisle, my teacher, for advice and motivation. I'd like to give thanks to Mr. Mel Brown, principal of Butler High School, for providing this class for me, Mr. Jack Andronka for mentoring and advice on how to exactly go throughout this experiment. And I would also like to thank the Congress of Future Medical Leaders of America for allowing me this opportunity for a scholarship. Here are some of my sources that I use. As I am a junior, I plan to graduate in 2015.